Good morning. Welcome to this year's distinguished lecture presentation. It's truly an honor to host and introduce Elias Zerhouni. He's had a really distinguished career, and just to give a very small overview, so he was born in Algeria, obtained the training there, and then had the majority of his academic career at Johns Hopkins University and Hospital. He's really a very, very successful researcher. He's a world thought leader on MRI. He was a pioneer on magnetic tagging of images to track, for example, how the heart moves, and refine CT densitometry to discriminate between cancerous and non-cancerous nodules in the lung. He has over 200 publications, 8 patents, and 5 startup companies. So remember that he's really done all the pieces in his career. In addition to being an outstanding academician, he was chair of the Radiology and Radiological Science Department, Vice Dean for Research, and Executive Vice Dean for the School of Medicine. After that, if that wasn't enough, in 2002, he became the 15th director of the NIH. And I have to say he has been one of the most impactful directors of NIH. Several of the accomplishments, briefly, is he launched the NIH Roadmap for Medical Research in 2003. He established an office to improve trans NIH initiatives across the institutes. Before that, they were very siloed. This allowed a lot of cross-collaboration. He also pushed forward the NIH-wide research initiative to address the obesity epidemic. He supported the NIH Neuroscience Blueprint. He ensured public access to NIH-funded research. So all the NIH PIs, I know you love putting your publications for access. You can thank him for that, facetiously and in earnest. And he was a big proponent for the convergence of physical and biological sciences, and I think we certainly have benefited for that here. So he stepped down as the NIH director in 2008. He told us that he was, for a brief time, scientific envoy for the president, which sounds very cool. And then he joined Sanofi in 2009 as a scientific advisor to the CEO, and was appointed the head of global research and development in 2011. And as many of you know, Sanofi is a global leader in healthcare. They have over 100,000 employees providing healthcare solutions in over 170 countries. Last year, they had 40 billion with a B in revenue, so that's pretty good. And he recently retired from Sanofi to pursue other interests. He has many awards, but has served as a senior fellow of the Bill and Melinda Gates Foundation and the board of trustees of the Mayo Clinic and the Lasker Foundation. He was elected into the Institute of Medicine of the U.S. Academy of Sciences in 2000 and received the prestigious Legion of Honor Medal from the French National Order in 2008. He was elected as chair of innovation of the Collège de France and elected to membership of the French Academy of Medicine in 2010. So with that, please, Elias, thank you for being here.
Thank you. Thank you, Andres. Well, thank you for this kind introduction. Again, I think Andres is very nice in sort of telling you about how an interesting career or bizarre career I've had, because frankly, going from academia to government to industry is not something that happens a lot because of chance. It was pure luck. But I've constantly been able or been lucky enough to be at the interface of multiple fields. My father was a teacher of mathematics. So in addition to what Andres will tell you, I'll tell you my personal makeup, if you will, that led to being able to, or wanting to do this, and to share with you the lessons that I learned. So the first thing was my father was a mathematician. He was a teacher of mathematics. And so I grew up actually never thinking that I would be a physician. He wanted me to be an engineer or a scientist, but not a physician, not a lawyer. Those are the things he said, you can do anything, but not physician, not lawyer. And so when I grew up, you know, we had, at home I had 6 brothers, and we were constantly challenging ourselves with brain teasers and mathematics and physics problems and all kinds of things that I loved. And you know, of my 6 brothers, 5 are engineers. So you can imagine the attraction there. But then one day I was a student, I went to volunteer in the mountains of Algeria. I'm from Algeria, from North Africa, so I'm an immigrant, I'm not born here. And I went to the mountains, and then we saw the misery of people in the mountains of North Africa about tuberculosis. And I saw an event that really touched me because it was a patient. She had tuberculosis, and she went to a medical center, and they didn't have anything because this was after the war. And the guy had you know, these old ice box where you put the ice, and that was to keep the vaccine. So he opened the ice box, and he said, put your chest here. She had, you know, 42, you know, her fever was probably 102, something like this. And she just went against the ice box, and she got a cold feeling, and she felt good. And then he closed it and said, well, that was the x-ray. And she said, oh, thank you very much. And then he gave her a shot. I don't know what, streptomycin maybe, and she left back to the, I was really touched by that. I thought, my God, there are better solutions that we could do. So for whatever reason, I decided to go into medical school. I thought it was more human than engineering. So, so I did. And then my father was really upset with me. I mean, he said, well, you're lowering yourself. Medical school is just basically rote learning. You're going to learn recipes about drugs and this. There's no intelligence in medical school. And so I did go to medical school because, you know, when you're young, you'd like to not do what your father wants you to do. And then you're always worried that you won't do as well as he did. So I did. I went to medical school, and I found out that he was right. After 3-4 years, I was bored to death. And it was boring, and you couldn't have a conversation with a teacher that meant anything from the point of view of science. And it was so empirical, all observational, there was nothing quantitative. And I always tried to put whatever they said in an equation system, and I couldn't. And so I was extremely frustrated. I was going to quit medical school. I said, I made a mistake. I don't want to go to medical school. And then a radiologist showed me the first CAT scan image. He said, look at this. And I was a medical student then. And he showed me these very big pixels, you know, you barely recognize it was a human head. And you could see the ventricles. And I said, how does that work? He said, well, you know, you have an X-ray tube, turns around, and they reconstruct with Fourier transforms. And I fell in love with that idea of combining physics, mathematics, and biology. So I decided that I was going to do imaging. And that's when I decided to come to the US to do research, and I was lucky enough to end up at Hopkins. And the rest is basically what Andres told you. So today, at the end of that, I wanted to thank Bob Neeram for inviting me and Andres to come here and asking me to speak about, okay, what did you really learn? What do you remember that we can use? So that's the topic of this lecture today. So I think when you look at any system, you have to really take a different altitude as you look at the system. So I call it the 100,000-foot view. First, never look at something from inside out. Don't look at your lab and then what else is there. Look the other way around. Say, what is the total system? And so what you realize very quickly is that academia, industry, and government truly are an ecosystem. They're separate, no question about it, but their agendas are complementary. And if you haven't really been around all three, you don't get that. You don't perceive that. So I'm going to try to share with you why that is and how you can navigate that ecosystem both as a student or as an institution. So the first thing that you have to understand is you go down to 10,000 feet. Now that you've seen the government and all of that, you say, okay, let me understand each component of the system. And we're engineers, right? So the first thing, you have to understand academia. And when you look at academia, you have to say, okay, what are the fundamental drivers of academia? What do we do really? Who are we? And if you get that, you then understand what are the connections to the rest of the system. I would say the number one role of academia is to be the guardian of the intellectual heritage of mankind, right? Everything that was learned over thousands of years is our heritage. And transmitting that heritage is one of the most powerful evolutionary force. Because frankly, natural evolution is basically based on transmitting information through DNA mutations, right? And we've done that pretty well for millions and billions of years. But knowledge, intelligence have allowed a different way of accelerating evolution by being able to transfer knowledge through education. So you don't have to have it in your genes, you just go to a great school like Georgia Tech and you get that evolution power behind you. So that's one. So the transmission of knowledge, the memory that we have of it is a key fundamental aspects of academia. Obviously, that leads to creating the human capital that any civilization, any society needs to be able to influence its environment. And that's what I think academia is also important as a social escalator. You know, anybody will tell you as you look at tables of compensations that if you have a high degree, you tend to be more compensated if you don't. And if you have a strong education, you tend to do better socially than if you don't. So these institutions are very key to the social balance and the evolution of society. But the fundamental drive is to understand the world. So whether it be basic or early applied research, that's what we do. That's what we want to do. We want to have the young capital of society come and really try to break barriers in knowledge. That's what academia is all about. But academia is very resilient. So here I have a 500 to 1,000 year cycle. And the question of 500 to 1,000 years came from a study that was done by a gentleman named Norm Augustine. And he looked at the history of governments and academia and industry and said, what's the lifetime, the average lifetime of an institution, of an academic institution as opposed to another? And you find out that if you look at the 100 universities that were existing in 1500, and you look today, 75% of them are still alive and well. And you can see that the lifetime of a university is in the 500 to 1,000 years range. Oxford, 1200, and so on. So they have a long cycle time. Very, very long prospects in academia. What about government? So if you look at government, you have to also say, well, okay, so what does the government do? Why do you have a government involved in the research that we do? Because it's based on a national vision and a national motivation. So the Kennedy speech for going to the moon was a vision motivation speech that really led us, led this country, to become one of the leaders of science, right? And to understand the power of that, the government has to espouse a vision. It's more or less good depending on the period you're in. I won't comment about now, but I comment about John Kennedy. So the second thing that you do as a government, you allocate resources to your priorities. So funding for NIH, funding for research is one fight that you have to fight as members of the academia, because if you don't, then the allocation of resources by the political system will be insufficient to sustain the dream that you see on the left side. So that's the second piece that you have to understand. But understand also one thing is that when you allocate resources to national priorities, like I was when I was at the NIH, I was arguing, our voice as scientists is the least potent. Least potent. Hear me out. We don't have as much influence as we think we do. You know who has influence in making the NIH budget strong? The patients, the ultimate customer, the ultimate voter, the ultimate person who benefits from the public point of view from the investment. And so that's what I think we scientists always forget, is that we think that it's enough to be rational and explain a good thing to government, and government will bow down, yes, doctor, and do it. My experience in Washington was different. We had 475 interest groups, Alzheimer Association, the cancer groups, and on and on. They were a lot more powerful than the National Academy of Medicine, the National Academy of Engineering, all of the scientific bodies. They were important, but they were not. So science is sufficient, is necessary, but not sufficient. All right. So you have to know how to build alliances, and the governance system, whether it be peer review or the congressional process, is a responsibility of government and regulation. FDA, all of these institutions. That's what governments do, not academia, not industry. And the public interest is really what drives the fundamental policies of the government. First, defense in general, health, education, the social needs, entitlement programs. And if you look at the debates, when you listen to the debate, if you can listen to a debate right now between, it's more like a foot fight, but, but it's not a debate. But whatever you listen, you see that it rotates around that. Some people want more defense spending and defense research, and other people want more social programs. You see that. And then if you ask yourself, how long can a government really last? And historically, are they long-lived? No, they're not as long-lived as universities. So the only two governments that have lived for 200 years or more, unchanged, are the UK and the US. They're the only two. Every other country has had a change of government through revolutions or other forms of upheaval. So governments don't last that long, and they have to evolve with that. Then you have industry, and you say, okay, well, what drives industry? What are the key drivers at the 10,000-foot level? The role of industry is to assemble the human capital that's created here to the physical capital, the infrastructure, the plants, the labs, whatever they do, and the financial capital. And the financial capital comes from multiple sources: pension funds, the street, Wall Street, investors, and so on. But that financial capital is the way by which government and societies allocate resources to what they think is valuable. And so the financial capital that you see here is allocated by a system that requires you to fill market demands. Nobody's going to fund industry if there's no demand for the products that they produce. But you have to do it such that the return on capital is sufficient to justify the investment. Do you understand? So let's just not dream. If you think industry, people have this dark side of industry, that industry is really a bad thing. It's not. It's really part of the ecosystem, part of the economy of the country that we need to understand in details. But what do they want to do is always under regulatory and competitive pressures. So I was talking this morning to some of the faculty, and it was interesting to see that sometimes when you look at, for example, the cell engineering manufacturing project, how much regulatory plays a role in terms of decisional processes within industry. Why? Because the chief of manufacturing in the industry, I can tell you, their number one fear is that the FDA comes in and stops them from doing something because they've changed the process. Changing the process of manufacturing in industry is the nightmare of the head of manufacturing. If you think they're, pro-change and pro-technology and pro-innovation, no. They actually want to keep things stable so that they can access the market because of the regulatory forces and obviously competition. So those are major, major issues. So in government, you talk about public interest. Here we talk about transmission of knowledge and creation of human capital. There they talk about regulatory and competitive pressures with what they have. And they're driven, obviously, to applied science and technology. They're not driven to basic research. We're driven on basic research here. So if you look at the lifetime of these companies and these industrial entities, it's much shorter. It's 25 to 75 years. Look back at the companies that you had around the country. For those of you who have our age range, so look how many companies disappeared. They're no longer here. And even companies like General Electric, which have been around for 100, is in great difficulty today, and a lot of people predict that it might actually disappear. And on, Westinghouse, and a lot of big names that you knew before have gone away. Why? Because it's really hard to maintain this regulatory and competitive pressure with a sufficient return on your investment. And if you don't, you go away. So there isn't that sustainability in industry as you do in, and it needs to be refilled and replenished with Googles and Amazons and then the next wave and the next wave. So that's why. So then when you've heard that and you've sort of extracted what the ecosystem looks like, what I'd like to do now is talk to you about the 1,000-foot view, about bioscience. So we are bioscientists. How is this evolving in this ecosystem? How is it evolving? What's driving bioscience today that you need to be conscious of? I think I can summarize it in 2 words: complexity and precision. So you say, well, what does he mean by that? And I'll tell you that there's a tension between the enormous complexity of biological systems. You know, in 1970, Bob remembers, there was a war on cancer. And the dream was that we would come up with a magic bullet to treat cancer. People thought in those days that biology was simple enough to be accessible to a single target, single bullet approach, magic bullet. Well, we know today that it's not the case. At the same time, because of this complexity, because of the stochastic nature of cell replication and the way things go, each one of us is a very, very unique individual. I can guarantee you your biochemistry is not the same as your neighbor. Your fingerprints are not the same. Your iris is not the same. Even your neurons, by the way. If you look at your neurons and you do single-cell sequencing of neurons, you find actually a lot of diversity in your DNA, between your own neurons, let alone between 200 different individuals. So just like your fingerprints are unique to you, your brain circuitry is probably unique to you as well. Statistically, it's almost having 2 human beings being exactly the same is as unlikely as winning the Mega Million lottery. You know, it's actually much more unlikely than that. So that is the resolution of things. We need to characterize the individual, but then we need to characterize those individuals in the context of a very complex system. So those are the 2 major poles that I have in my mind. And then when you understand that, you understand that there is no chance for us to succeed unless we understand that we need multiple disciplines coming together to really crack these problems. I mean, you need quantitation, you need new technologies, you need a culture of cross-disciplinary collaborations, and you need structures to do that. And that's one of the things I did when I was at Hopkins. I created institutes without walls, because departments tend to put barriers to interdisciplinary work. And at the NIH, if you look back, you say, you know, it's been impactful. The number one thing I did was to create open doors to physical sciences, biological sciences, and multi-PI grants, which allow interdisciplinary work. And the convergence between the physical, the biological, social, and data sciences is obvious, it's growing, but it has profound implications on your career. So I'm talking to the students here. This is not empty talk. This is something that will impact you. And you have to understand it, and I'll tell you what the rules of the road are at the end of the speech. The other thing is we have a huge failure rate. I mean, I was shocked, you know, when I took this job, I thought I was going to look like a hero, you know, like I was so smart that I was going to get drugs done and treat Alzheimer's disease in 2 years. Well, wake up. The work of research and development is beset with failure. You have to manage failure, not success. Success will come if you're lucky. Failure will be there for sure. It's the normal state of affairs. So you have to really understand that the knowledge is insufficient at this point. The knowledge is insufficient at this point for us to be predictable and predictive of the success of any scientific approach, which means that you have to create the opportunity for people to try and to do new things. And that's why I think basic research still remains, to me, the priority of funding by the government. So if you look at medicine, and I'll take you there for a minute, if you look at medicine, traditionally, what have we done as doctors? For years and years and years, we waited for the patient to be sick, and then we looked at them. And you had Galen, all of the Aristotle, all of the ancient doctors who basically had these ideas, and they formed ideas about humors, fire, heat, that they observed from the outside. They went from the outside in. And then you had organs, and then Harvey discovered the circulation, the heart, and all in all, it was really an outside-in exploration. Today, in biomedicine, the world is inverted. We are going from an inside-out exploration. So what do I mean by that? We've become very cell-centric and DNA-centric and molecularly driven. So when you look at the way we look at the world today, think of concentric circles, and DNA is at the center, and then RNA, and then proteins, and then the cells, and I'm making it simple here, which then organize themselves in tissues and organs, and then they organize themselves in integrated organisms, which are interacting with behaviors in our case, because we do have emergent behaviors due to all these changes, which are also driven by the environment. So all these layers of complexity and the multiple potential permutations of this complexity also drive, this complexity drives the individuality that every one of us is unique. Whether it be at the DNA level when we do genomic studies, or at the RNA level, the protein level, whatever it is, I mean, at some point, you'll have a divergence that occurs within your own development to make you different than anybody else. Does that make sense? Okay. So if that makes sense, then you say, okay, fine. How do I then play my cards as an institution like Andres needs to focus on making you guys successful and you being successful? And the way I look at it is think of it as a multi-layered network system. So from the engineering, I don't know, network guys here, but basically it's a multi-layer interacting network. So if you look at the DNA in the first place, the DNA is interacting with its transcription machinery, with its epigenomics, and then interacting with the RNA layer, with the protein layer, and I'm making it simple, and the metabolic layer, and the cell and the environment. So that's what it is. So when you have a mutation in the DNA, that mutation is affecting a network. And if it's a mutation in a molecule like C, which represses A and encourages or represses E at the end, what you end up with is a network where A and E are going to be overexpressed. Does that make sense? Right? The total thinking of this approach is called systems biology. So people are trying, and in many, many different fields, yours included, to truly template that. And so it's driven, obviously, by epigenetics. If there's methylation, acylation, you don't know. The genetics and the genomics, the transcription, the translation, regulation of all of this, and the signaling of molecules across, and then obviously proteomics, and this is just one module. You can imagine this being immunology maybe. And then you have to really see what, at the metabolism level, this whole thing means. Do you have the balance that you need to have in a normal or in a patient, let's say with diabetes? And all of that is essentially leading to emergent properties that come in from each one of these networks to the next one, to the next one. And it's really hard to invert that, to understand that in inversion mode. Even when you study the neural system, EEGs are a good way to get signal, but you can't invert that into understanding how the brain works. So we have multiple levels of doors that you can only traverse one way. So having said that, the environment is very key. We are not evolving in the vacuum, we're evolving in the environment. And so you can never forget that at any one time, any one of these networks can be affected by an environmental factor. All of that is those are the elements of this complexity that I talk about. And you need to understand that not everything is driven by genetics, because there's a sort of gene-centric view of the world here, that everything is in the mutation that you can find, and that's not true. It cannot be true. and the environment drives a big role. Actually, to prove it, I'll show you an experiment we did. We had the statue of David that has no DNA, moved over to the US, and this is what happened after 1 year. That was, so that was the proof, positive, that the environment does play a role. As you well know, we do have an obesity crisis in the US. But anyway, so when you do analogies, you try to do analogies. It's a little bit like if you were an extraterrestrial scientist 1 million years from now, and you discovered the chip on the right side there, and you try to ask yourself, how did that work? You could probably decompose the chip in its components, you know, transistors and all this, but you will never understand how it did, you know, a PowerPoint presentation or a work perfect document. You never understand how the software was, unless you have a totally functioning system. So reductionism of taking things apart is not sufficient. You need to really reintegrate. And if you look at biological sciences, we are right at the knee of that effort, where we're trying to reintegrate the components into an understandable whole. And that's what cell networks, this is a cell on the left side. These are the biochemical and signaling pathways that you can find. And the complexity is probably greater than reading a chip from Intel, as we could imagine. So that's what I call the era that we're in, is the era of quantitative. Remember the word quantitative. There's no science without, if you can't measure it, you can't manage it. And it's a network type of biology. So having said that, you say, where are we going? We're going really from what I call the hardware periods of science, where you take the components and you try to understand each component and how they work internally, an ion channel, a transcription factor, and whatever, and you're trying to put them back together into a coherent system. Actually, Bruce Alberts uses the word, the cell is a computer. If you listen to him, we've had this conversation many times, he believes that cells are fundamentally computers. And they're computing multiple inputs to create an output across multiple networks. So for him, understanding the software is as important or more critical than understanding the hardware today. So that's what I think the molecular networks, the disease pathways, the regulation in health and disease, that's the core of what we do. That was the core of the roadmap. When we did the roadmap, we said networks and pathways, scientific teams of the future, which have to be multidisciplinary and really hardworking together. And that should lead to a reclassification of diseases. And if you can reclassify diseases to their mechanism, you can have biomarkers in immunology or somewhere else. You can understand the drivers of health. And more importantly, then you can start to have AI play a role in understanding the complexity across multiple individuals. Because that's the issue. The issue is one sample of one does not tell you what the truth is, but sample of many without individuality also doesn't tell you anything. So those are the factors, and that's what I say to people. You know, when you look at disease networks, and I'm just drawing this one, you can discover one, but some molecules are more important than others. They're hub molecules. And so the idea is to really do multi-targeting, where you could essentially act on this molecule here, which will affect this one, these two diseases and this one, and then you can combine it maybe with this one or this one, and you have to decide which ones are going to be the most important. You know what I'm describing here? That's what's happening today in immuno-oncology. Everybody in immuno-oncology is trying to figure out the next partner to a PD-1 checkpoint inhibitor. That's the science that is exciting a lot of people. It's no longer the science of the tool, it's the science of the system. So you can have an RNA or a small molecule or a gene or cell therapy like you're doing here, or mono or multi-specific antibodies, peptides. The idea is multi-targeting of coherent components of the network that can affect the disease process. So that's where we are. And I'd like to just give you a quick example of my own field, because I'm talking about things that I don't quite know, except for imaging. That's my field. I know that. And I can tell you the impact on imaging that this whole revolution has led to is truly something that I think I can share with you, because before we were imaging people, now we can imaging cells, molecules, but also populations. And I'll show you examples of that. But I think the key thing for those of you who are in imaging at all levels is what's the definition of bioimaging? So here's my definition. It's extracting spatially and temporally resolved structural and functional biological informations from molecules to humans, right? That's my first work, was actually to try to characterize cancers from benign disease by measuring calcium content. And then I transfer them to osteoporosis. That was my first startup, was to help people treat osteoporosis and prevent surgery for benign disease. So these are the kind of things that essentially drive imaging. It's different than a destructive technique where you separate components, you don't know where they came from. Here, this is spatially localized. So what are the two things that are driving imaging sciences today? I'll share them with you. One is the revolution in molecular and cellular imaging. Because we understand better the biological targets with antibodies, with other molecules, we are able now to image things that we didn't think we could image even 5 years ago. So an example is when you develop a particular molecule for the human body, you'd like to know where it's going, what target is it attacking, is it there in sufficient concentration for you to do something about it? These are the fundamental questions that any scientist will ask themselves. Today you can image that. You can image that with long-acting isotopes, because you need to localize and assess the functionality of these molecules and their targets in vivo. You need to characterize and quantify the disease-specific patterns. I'll give you an example. And you have checkpoint inhibitors, right? Very expensive. But 50% of patients don't respond. 75% of the patients don't respond. Why not? Is it because the target is not there? And you would like to know if the target is there, right, before you spend a huge amount of money on this. That's a key question in immunology and oncology. Why is it that this is happening? And so multimodality imaging for biomarkers is the revolution that's ongoing. So if you look at, for example, this study that we did at Sanofi with a partner in the Netherlands, is a zirconium-labeled monoclonal antibody that we developed against TGF-beta. Now, TGF-beta, you know, is an immune suppressor, so we wanted to suppress the suppressor. And, but to do this, you have to find out if TGF-beta is overexpressed in the target. Because if it isn't, you're going to have side effects with no benefit. And so by doing this, we showed that the tumor here was actually hot in terms of TGF-beta. And that's in humans. That's not in animal models. It's directly in humans, and it really helps you guiding the therapy. The same experiment here, with different tools. This is the PD-L1 antibody against the PD-L1 target to find out if the cancers that you see in the liver here and the lung are actually active in PD-L1, so you could use a PD-1 monoclonal. Again, this is PD-L1 in non-small cell lung cancer with a nanobody. So this is a monoclonal, 150 kilodalton, very large. It doesn't go everywhere. Nanobodies, this is from a company in Belgium that developed camel antibodies from camels and llamas. And they're very small, but they're very specific. And they target very well. And then you have other molecules, 14 kilodalton, or peptides. Why is it important? Why is that important? It's absolutely critical, because you will not be able to permutate all the experiments you want to do without having an inhuman biomarker, right? Otherwise, we're going to treat people with 5, 6 drugs at huge cost, not knowing that they're efficient. So here is a study by one of my students, Marty Pumper at Hopkins, who found an agent that he could target against the prostate-specific membrane antigen. So that's an antigen that is specific to the prostate. And he was able to find a way to label it with fluorine 18. And if you look at the experiments, it does it in 30, 60 minutes, which is way within the half-life of the isotope. And look on the left side, this is a patient who basically perhaps had one lesion here, but these are rib fractures, and basically you could have told them, you're doing very well. Your cancer is actually under control. You have a single lesion, we're going to radiate that, you'll be doing fine. But look what happened when you did the specific imaging with the PSMA. This patient was riddled with metastatic disease. that we didn't really see in the previous test. So that's important. Here's another aspect which would be of interest to you because you're doing CAR T-cell manufacturing. So Marty had this idea, again, Marty is a very creative guy. He had this idea of, when you do a CAR T-cell, you put an antibody that targets the tumor, right? So he said, well, let me put the PSMA, let me put the antigen that targets his marker on the same CAR T-cell. So he did a CAR T-cell experiment where he had the targeting of the tumor and the targeting of the imaging agent. Do you understand? And so what he shows, he said, because the big question in CAR T-cells, are they there? Are they going to the tumor? Are they doing what they're supposed to do? And so he showed that actually this is after day 12, he was able to show that these cells actually were infiltrating the tumor in the mouse model. So that kind of revolution, I think, is going to grow, something you should look at. A lot of departments are not looking at that in BME and imaging departments because you need cross-disciplinary collaborations with chemistry, with radiochemistry, with nuclear medicine labs. So you should do it. The second revolution that I think is important is what I call radiomics. And it's the machine-augmented diagnostics. And I think it makes a lot of sense that we're going to see AI and machine learning methods classify these individual patients across huge databases. As a radiologist, when you look at an MRI of the brain, you compare it to your own memory. But your own memory might be 5,000 cases max, 10,000. If you're really, really, really good and work every day, you may have 15,000 cases in your mind, most of which are probably unproven and you don't know what the study is. And you don't really remember what the previous study was, and then you pull it, and it's messy. It's messy. Except for local disease, we don't really know how to read diffuse disease. And so if you want to track the evolution of a disease, you'd like to compare it to some database, right? And these databases are going to become the money of the realm, like they say, the gold mine. Because if you are a system, you can put 5 million patients, MRIs of the head in the database that you follow over years. Look how important that will be. When I read the scan, I'll put the scan in the machine, and the scan will tell me this patient is like patient XYZ in that database, which have this disease, that disease. Can you imagine the power of that? 10 years ago, you couldn't even have dreamed of doing this. We did it for mammography 20 years ago, but we couldn't do it for the rest of the body as we do today. And so that information, machine learning, is going to be fantastically critical and important, which really basically you can apply the exact same thinking to genomic data, to genetic data, to single cell sequencing data, you name it. Any big data scale can be approached now with these integrated reference databases that will teach us about populations much more than they will teach us, and it will help us manage individuals. So this is, for example, the dream of all folks in imaging is to segment the image and understand it. So this is manual, and this is by deep network prediction. It was done by Elliot Fishman with an NVIDIA platform. Amazing power. It's almost like you don't need to be there. It outlines the organs for you. And the nice thing is you can use that to characterize tumors. And you can say, well, here are liver lesions. There's 1, 2, 3. They're different, right? And here there's a very vague one. The machine actually can extract them and analyze the texture of it to the point where it can actually distribute through these parameters that come out of deep learning and separate what we call focal nodular hyperplasia, hepatocellular carcinoma, a benign tumor, or a normal tissue. So you can see how the power of these three-dimensionally spread parameters, which frankly, I don't know how they came up with that. I don't think anybody knows how the algorithm and the neural network did it, but it did it. So you can imagine now that if you want to treat a cancer patient and you have these sort of imaging that are routinely achievable today. This is a patient with, you can see the heart here, you can see the liver, you can see the stomach with full fluid, you can see the vessels going into the liver, and you see all these lesions, right? This is a patient with a carcinoid tumor which invades the liver. You know, I would pay a fortune if I'm really trying to understand and treat this, to know very quickly, in a matter of days, not weeks, not months, if the therapy I'm using is working. So how do I do this today? Basically, you take a ruler, you measure 1 or 2, and you come back, and this is what we call the RECIST criteria. Ask oncologists, they'll tell you. When you hear about this RECIST criteria, you can't resist laughing, because it's so, but that's the only thing that is working today. That's what the FDA approves. So you have to know that to know the power, potential power of this. Now, when I was at the NIH, we put an initiative, and Bob probably remembers that. This is an initiative between the NIH and the industry to understand Alzheimer's disease. We started that in 2003, and we had MRI images, LCM, CSF sampling, and the need was to follow these patients. And Dr. Thompson at UCLA had a really brilliant idea where he would indicate the variation within the brain of patients. So green means not a lot of variation, blue, a lot of variation in patients between Alzheimer's patient and non-Alzheimer's patients, right? So he was able to sort of use these indicators. But the next step is not that. The next step is going to be what I said. In other words, we're going to create huge databases of normal development of the brain. This is a study that Judy Rappaport started at the NIH in 2005 or 6 of trying to understand normal brain development. So she would take images with MRI, with diffusion tensor imaging and all this at sequential times with these children and follow them over years. So it's like a Framingham study of the brain. Framingham was the study of the heart. This is the study of the brain. And over time, hopefully, we'll have reference databases for normal development across the ages, all the way to 85 and 90, and see if we can identify as early as possible predictors of degenerative disease or Parkinson or any other disease that's affecting us. And I think that's going to revolutionize that, because imagine if you could do this for all human brain studies and put them in the cloud and have them and compare that. The power of that on biomedicine is going to be a revolution. So I think I've made the point, because it allows us to be more precise and more predictive. Imagine, for example, you do a colonoscopy now in patients to prevent colon cancer every 5 years or 5 years. Imagine if you could use one of these imaging scanners now. In 2 minutes, you can get the entire body. I mean, it's amazingly powerful. And let's say you had that as part of your physical every 5 years, just like you do your blood test and your cholesterol. How powerful would that be in terms of detecting disease, in terms of if it's combined with circulating DNA, liquid biopsies, or anything like that? You can imagine a world where I think all of us as individuals will have the ability to be compared to normals and abnormals to predict what we need to do or not do to treat our patients. So is that a pipe dream? I don't know. Sometimes people say, you're crazy. It's never going to happen. It's too expensive. It's not true. When I did the CT scanning at the beginning, people thought I was crazy because these machines were costing $2-3 million at the time. But today, you can get a scanner for $300,000, and you can do more patients than ever, to the point where there are companies that are offering this sort of test right now. So it's not a pipe dream. Now, let me finish, if I could, by some personal advice to each of you. I'm talking to the students here, Bob, not you. You don't need any advice. He needs advice. He needs. So the first thing that I would say is I have some rules that I'd like to share with you. First is 50-50 rules. What does that mean? As I was doing what I was doing, I realized that I was very curious about things beyond my own specialty. So when I was reading radiology and imaging, maybe 50% of my reading was radiology and imaging. But 50% of that was actually non-radiology, non-imaging. I would read about chemistry, I'd read molecular biology, I had science, nature, things that I barely understood. But over time, it made me richer in understanding what I could get help from. So 50-50 in your friends. You know, I had friends in medicine and radiology at BME, but I also made friends in other parts of Hopkins and outside of Hopkins. One of my best friends is actually an accountant. I mean, it's hard to believe, but it's true. It's actually useful for taxes, but... And the only rule that I wouldn't say 50-50 is when you're in love, it's got to be 100%. Don't, I mean, at the risk of your life, you don't miss. mess that up. So that's first rule, and you understand, enrich yourself in areas adjacent to who you are. The second is always define a core strategic theme for yourself. Now that's a hard one. That's a hard one. But follow me a little bit. And what I found, but this is retrospective, I mean, I learned it after the fact. I didn't really know it at the time, but I think it's a great lesson that I learned between all these fields. If you really look at my career scientifically and you say, what's the core theme? What is the strategy you followed? What is the theme that you were after? I would say that my big driver was to bring quantitative sciences to the qualitative biological sciences as I saw that. So I could tell the radiologist was looking at things and making judgment with no measures. And I said, that's enough. That's why I created CT densitometry. I created cardiac tagging to measure cardiac function. We looked at MRI signals for tumors. But it was the same idea. It was, how do you quantitate what really happens in a biological system so you can make sense of it? Then, when I was in NIH, I was a little more mature, so I knew what I was going to do. Because when I was asked to do the NIH, I had the same problem I had at Hopkins. Departments were not talking to each other. I mean, Don Giddens is here, he knows. I mean, we had BME, but really reaching out to Don and at electrical engineering, computer science, mechanical engineering was not natural. So we forced ourselves to do it, and then we created these institutes without walls. And so when I went to the NIH, I have to say something that makes a lot of sense to everyone, right? The core theme. And the core theme idea that I came up with was focus on what NIH should do. NIH is an institution that no single institute could do. That was the exact same thing I did at Hopkins. I kept telling the department chiefs, well, is there anything that you would like to do? Yeah, I'd like to do this, but I don't want to do it. I can't do it, don't have the resources. And then when you surveyed the faculty, every one of them would say, well, we need a core of this or the core of that. So things that the institution needs to do, but that no component of the institution wants to do or can do. So that was the theme of the entire thing that I did. If you look back, everything came out of this concept. And when you ask the question, the people came together. The people at NIH said, yeah, and some people hated the fact that you had the directors were telling them what to do, but I wasn't telling them. I said, you decide. And once they really decided, they loved the idea that they could fund things out of a common opportunity fund. So that's what we did. You know, the structural biology, the network analysis, the teams of the future, the multidisciplinary teams. Now, in my last job, I told people, I said, look, I worked a lot because you can't have a strategy if you can't write it in one sentence or two. You have to have your own personal strategy to do that. You have to define yourself. So I said, you know, at Sanofi, we're going to understand molecular networks of disease and attack them with multi-targets approaches. That's what we did for the past 7 years. So the other thing that I can tell you is that don't think the future is not readable. It's readable. And you can tell what's going to happen in the future. I'm surprising you, right? Everybody says it's really hard to make predictions about the future, except in biomedicine. In biomedicine, things take a long time. So what I found out when I evaluated the NIH programs, I found out that when you looked at the medicine of 2003, 2004, everything had been there. The drugs had been there, imaging had been there 30 years before, except that it was not appreciated. So the key thing for you is, what do I need to do to appreciate what the power of something that is currently here will be in 10 or 15 years? And it's easier to do when things are going to be in 5 years. So we know bioprocessing is going to be very important in 5, 10, 15 years. AI is going to be important. So you decide what is really critical for you. And the lesson I always tell people is that, you know, surfing is a great sport. I don't know if you surf around. You probably don't surf around here. But anyway, if you did, you know that when you surf, the thing that makes you great is not your skills, is the wave that you pick. So pick the right wave that will drive you to a future where you have a core theme strategy that applies to the 50-50 rules. Why is that? Because I will leave you now with the last concept that I believe is important today, because everything I said has an implication on how you develop yourself as a student. It's completely different than it was in the past. So in the past, if you went to a discipline, you developed an expertise. So think of it as a vertical thing. You go down into your silo, deep, and you're really, really good, and you get a PhD, and you get a postdoc, and you stay in the same lab, and it's great. But the problem is that science has evolved from a reductionist era where you needed to do the components of systems to an integrational era where you need to reintegrate the systems. Well, you can't do that all by yourself. So you could do and say, okay, I'm going to go lateral and collaborate with everybody out there. So you could say, oh, my strategy is going to connect. I want to connect to people, connect, and be a connectivity guy who is very shallow in everything, sort of jack of all trades and master of none. That's like salesmen. Salesmen are like that. They know everything, and they can tell you about the car and so on. You don't want to be that either. So what is it you want to be? You want to be this, a T, a balanced T, where you know how to connect laterally, but you have a grounding in what you do. So for me, I'm grounded in imaging. I really believe in this science. I know that science inside out. But also connected, like I said, because the 50-50 rules, and I'm pretty good at other things. I can fake it most of the time. And, but that makes you richer. And in the world of today, building teams through having this sort of design in your mind of being able to cross barriers and to remove barriers and to understand each other in different cultures and different environments is critical to success, because the scale of science today is different than what it was 25 years ago. So the key thing is that all of these are converging. The complexity, the precision, the diagnostics, the therapeutics into precision medicine, and never forget to have fun. Pick the right wave. Thank you very much.
That was great. Thank you. We have time for a few questions.
I'd love for your comments about, as we appreciate the complexity, the role of the environment, the role of the individual and population base, we're getting ourselves further and further away from causality and more into association. Can you comment about the strengths and the dangers of moving towards this? Because we can't possibly do all the experiments that are needed to define causality.
Yeah. So that's a very good question. It's a fundamental question, because when you do these correlations, it's correlations, and you can't really have a causation unless you do a validation experiment, right? So the key thing is this is a learning system that is essentially going to point you towards possible logical causation that you have to test in a statistically valid way for an intermediary period of time. There are some theoreticians that say that you can actually, in a large enough population, start to identify control groups. So you can do control, but frankly, from the statistical point of view, it's not pure. And you have biases in the selection of the population and so on. So I think deep learning in applications like the one I showed, where you're really following the same patient population over time, may have actually the power to identify the stronger and stronger correlations, because otherwise the correlations will weaken over time. Just like you do, like we were talking yesterday about genomics. So I don't have the answer to your question. I think it's a key question. 96% of observational studies prove to be either incorrect or statistically invalid. 96%. So everything you read I always find fun, People who drink coffee at midnight are likely to be dying sooner, or people who take milk at 12 live longer, I mean, that is junk. I mean, that's fake news. Yeah.
I had a question. I enjoyed your talk, and you were talking about the complexity and the value of interdisciplinary approaches to tackle the biomedical problems. My question is, what are your thoughts on the
So
So
if
what approaches do you think is the best to take in those scenarios where the same disease have lot of variability, heterogeneity? For example, even when you take an example of brain cancer, not all brain cancer gliomas are having the same pathways. They are so different.
Right.
So from a therapeutic point of
Right.
view, how do we approach that? Do we look more into basic science, looking at markers, or we go into directly patients and look at the changes in the tumors?
So step one is to reclassify what you call a disease. Because you call a disease a heterogeneous entity, it's probably not one disease, it's probably multiple. It's like dementia and Alzheimer. I mean, when you look at Alzheimer diagnosis, many of them don't have Alzheimer, they have something else. So the real point that I was making, if you remember, was a big effort that I think needs to be made is to reclassify diseases into their molecular phenotype or reclassify the heterogeneous cancer you're talking about. I mean, you know, breast cancer, we know already that there's a HER2 positive and a estrogen positive or triple negative and so on. So precise reclassification is going to help solve this problem to a great degree. The second is to study very specifically the non-responders. Beyond that reclassification, why is it that a patient doesn't respond? So in the European Union, I was pushing for a program that they accepted actually several $100 million to look at the non-response in autoimmune diseases. Because it's amazing. I mean, you treat patients with autoimmune disease and 30–40% of them don't respond or stop responding. And so understanding that heterogeneity is important because it's probably a source of discovery of new targets and new pathways. Last but not least is the signal-to-noise problem. And in the signal-to-noise problem, what you have insufficient sampling. And because you have insufficient sampling, it appears heterogeneous to you, it's grainy. And so you need bigger sample size. And so pre-competitive collaboration between different entities, different health systems to accumulate enough in any one disease is going to be very important to control the signal-to-noise issue.
Great. Thank you very much for the wonderful talk. I was really struck by your comment that patient advocacy groups have a lot of power, at least administratively, with the government, and how that contrasts with the traditional way we see patient-doctor relationship, which is very much doctor down to patient. And we talk about personalized medicine as changing and influencing that, patient more power over their own medical decision making. And I'm wondering how you see that relationship evolving and what the role of scientists is about it.
So from the patient point of view, you know, the thing that I think is very obvious is that as we're moving more earlier in the disease state, and we are able to prolong survival for many disease states, that the patient is going to have to play a more active role. So I used to call that P4 medicine. You know, it's more predictive, more personalized, more preemptive, but more participatory. So the intelligent participation requires a fair interpretation of the data, and this is what patient groups do, and they do very well. I mean, it started in rare diseases. I would say the best example is the Cystic Fibrosis Foundation. You know, 30,000 patients, they were suffering, they were dying very young. Today they die at 43, there's no cure. There are treatments, but no cure at this point. And you can see that they are participating and actively going into their care. The second point that you're making in terms of lobbying is that, you know, patients are everywhere and they truly believe in the power of research. And the good ones, they don't really, you know, you would think that they want you to do treatment experiments, you know, give me some stem cells. There's some of that. But most of them, if you look at the strategic plans now, the quality, and I help them with that. You know, when I was at the NIH, I said, guys, what do you want? You want informed patients or uninformed patients? And what is the best? And you realize that you have to participate and help them and participate in them. If you look at the Parkinson's Disease Association, you know, the Michael J. Fox, and you look at their strategic plan, they're better than many strategic plans I've seen in academia or even at NIH. So I think it's what I tell people, our best friends are not the industry, not the politicians, not the payers, not the insurance companies, they're the patients. And as doctors, we're losing a lot of that. there's a lot of intermediary bureaucratic decisions that are embedded now in the healthcare system, which have lowered the power of physicians to decide. And if you know about what we call authorization, do you guys know what authorization means? Pre-authorization? When a doctor prescribes a drug, there is a formulary that the doctor has to abide by. And the insurance company can deny treatment if you don't really do something else first. So for example, if you have high cholesterol and there's a drug that really reduces that, they say use the generic first, 3 months, then do a test, use the second generic, and go through that before you take the expensive drug. So they're practicing medicine for us. You see the same thing in children. My wife is a pediatrician, and she told me that she was fed up because she would spend half an hour with an unknown healthcare provider or coordinator trying to argue why because she does pediatric endocrinology, a 3-foot kid needs growth hormone. I mean, that's near puberty. And she was very, so we are losing that, not gaining in that, powerful relationship that you described. It's less powerful, which means that we have to really engage with our raison d'etre, which is patients, right? I mean, we're not here to do, in medicine, we're different here because of engineering, but for patients, you have to engage. And we don't. And you would be surprised how little, I'm surprised people when I say that, but you get some influence, maybe 10% of the influence, but a good 80% comes from patient groups. So you have to nurture them.
Right here.
Yeah.
So one of the key initiatives you as a political is advancing trends in research. So that led to creation of this award and remediation of NCATs. So I want to know the reason why you pushed that forward so strongly, and was there a difficulty in breaking the program, and what were the core factors in assessment?
I didn't understand the last two parts, the last two sentences. How is the program managed? So it started first by, I didn't do the roadmap out of nowhere. So what I did is I was appointed in May, and the NIH had a director for a couple of years. So it was really frustrating to Congress because Congress felt that they were increasing the funding for NIH, but they had no plan. So it's like me giving you money and you say, well, I'll tell you tomorrow what I want to do with it. And they were frustrated with that. They wanted a plan. They wanted an understanding. So what I did is, it was different than most directors do. I didn't come with preconceived idea. I had some. I mean, don't mistake it. I did have some ideas, but what I did was I invited 125 scientists from around the country, and I had joint sessions between the internal institute director, institute scientists, and the external, trying to identify just what I said. What is it that the NIH should do that no institute can do, right? And we came up with about 97 different concepts and ideas. One of them that came actually, you'd be surprised, but it came from the Nobel group, Al Gilman, who was consulted, Harold, others, who said, we have a problem. Greengard was a big advocate of that. David, Baltimore, I mean, they were basically saying, look, Elias, we are doing all this basic research, and you have all this clinical research over here, but nothing is happening in between. With the new emerging science, we need people who understand that and can translate that into the clinical. So if you remember, at that time, we had the General Clinical Research Centers. I happened to be a PI of one at Hopkins before I went to NIH, so I knew how it worked. It's basically nursing support for the investigators. And you have a lab, you have a connection to your lab and a pharmacy, research pharmacy. That's what it was. It was a service function. So what I think the folks were asking for was not a service function, but a human capital formation of folks who understood enough to traverse the valley of death and really establish degree-granting programs in translational science. The first one that was established actually was in cancer at Memorial Sloan Kettering by Harold on cancer science, you know, translational cancer science, because you do not see that. I mean, would you go to, if you have something here, who would you go to do a translational effort? You have physician scientists, but you know, they're not in the best of shape inside of academia. And you have, so they decide to go into basic research. 85% of MD PhDs end up in labs doing you know, animal model research. 15% ends up with the clinical part, the late stage, but there's nothing in between. And so that was the idea of CTSAs. Do you train people in new science? Do you really have them adopt and transfer the basic research they participate in and understand into a system that is completely different than the previous one? The hard thing was to destroy the previous program, because in government, you don't do that. Every program was in many institutions, 62 institutions. They had their own lobbyists. And the lobbyist, I remember her, I mean, she would look at me like I was the devil. You know, I said, oh, what did I do to you? She said, well, you're wrong and this and that. Imagine, I mean, if you haven't had that experience, you should try it once, you know. If you get the opportunity to go to government service, do it, just for the education of it. And so they didn't want that until I found a way to get it done with proper convincing of some Congress people. And so we switched that. The key thing to me is whether they're successful is what kind of people are they training who then become the translators between basic and clinical, which was the big gap that you found.
Really great talk, and I was particularly interested when you talked about large databases with machine learning and AI. And I'm wondering how you see that type of information being owned and disseminated. So I think it's a very disruptive approach to traditional R&D in the pharma industry. And the reference databases that you made reference to have been generally publicly funded. But the outcomes databases and supporting the pre-competitive analysis, how do you see government, academia, and industry, what are their roles in disseminating, owning, and creating these databases for the benefit of patients? And how do you see industry potentially responding or changing their models in order to be those profitable institutions that are going to create the new
You know where the big problem is? It's not in industry, it's in academia. If you really look at you know, the ownership of data, jealous ownership of data, it comes from academia more than industry. If you really, I mean, try to get data from a group, the Framingham group, I don't want to, but they consider that their lifeblood. Epidemiologists, it's their lifeblood. They don't want to give the data. So the highest level of resistance actually is in academia, not in industry or government. Government, as you know, we make it public as soon as we have it, right? The sequencing is a good example and so on. Where you see now accumulation of data is in healthcare systems. So the Partners, for example, in Boston has a 3 million database that we funded at the NIH that allows them to identify patients for purposes. The Geisinger Clinic. Then you have collaborations, pre-competitive on Alzheimer's disease, for example, between industry and NIH, osteoarthritis. So it's coming together. But frankly, it's haphazard. I don't think there's a systematic view. I mean, look, Optum, which is an insurance company, part of United, has 315 million health records worldwide. And they do what they call real-world effectiveness, which is the understanding of who is using what medication versus, and comparing that. So they're not doing correlation studies, they're doing comparative studies of efficacy and safety. So some people are accumulating that data already. It's going to be a huge issue of the privacy, the protection of privacy, and it's an ongoing story, but I don't believe that there's going to be a simple solution. There's going to be a case-by-case solution over time. Healthcare systems are probably the most likely to integrate data from their patient base as they're getting larger. So there will be an Emory database for sure.